The we have the speaker here today. She's Dr. Hager Kowash. She's a postdoctoral research associate at the University of Manchester with three research interests that include fetal programming, placental biology, and the developmental origin of diseases. So, after completing her undergraduate degree in medical sciences from the University of Leeds and her master's degree in reproductive and developmental biology from Imperial College London, she completed her PhD at Manchester, investigating how stress during pregnancy eliciting maternal immune activation response that could lead to the propensity of offsprings to develop neurodevelopmental disorders such as schizophrenia in later life. So now, as a postdoctoral researcher, she is interested in developing potential dietary therapeutic interventions to treat pregnancy complications such as fetal growth restrictions in mouse models, working initially on nitrate and beetroot juice, and now currently she's characterizing and developing potential therapies for pregnancy complications using EVs derived from fruit and vegetables, in particular watermelon. So today's talk is about the impact of extracellular vesicles derived from watermelon and placental function and pregnancy outcomes in an in vivo models. Over to you, Hager.
Yes. I will just share my screen 1 second.
Yeah.
Okay. So thank you very much for the introduction. Everyone can see that okay, I'm assuming.
Yes.
So,
Yes.
yeah, thank you very much for the invite, and I'm excited to talk to you today. You've already kind of gone through my introduction, so I'll just whiz through this now, but just some pretty pictures for you about where I've been and how I've got to where I currently am. So, so I started off at the University of Leeds for my undergrad, and then I found that I had an interest in reproductive sciences, so I did a master's in reproductive and developmental biology at Imperial. And then I worked as research assistant slash technician, firstly at the UAE University in Abu Dhabi, working in the paediatrics department on various different projects. And then something very different as a research technician at the University of Sheffield, working on pulmonary hypertension. But I always realised that my passions lied in maternal and fetal health. So that's when I went to Manchester about 7 years ago now, and did my PhD on the maternal immune activation model and trying to investigate the role of the placenta in mediating developmental programming of neurodevelopmental disorders, as Dalmina mentioned. So now recently all my postdoc work has been on developing dietary therapies for pregnancy complications. Firstly, working on a high-fat diet, obese mouse model, using beetroot juice as a source of nitrate to potentially rescue the phenotype of FGR. But the last 4 years have been focusing on extracellular vesicles from plants, fruit and veg, as potential dietary therapeutics. So that's what I'll be talking to you about today. And we're specifically talking about watermelon extracellular vesicles and their effect on the placenta in a mouse model. So I'm sure this audience is very familiar with the placenta, or maybe there may be a few people that are not so familiar with it. So I'll just do a quick recap on the importance of the placenta. So we know the placenta is a transient organ in pregnancy. Its principal function is to supply the fetus, especially the fetal brain, with oxygen and nutrients, but it's also involved in fetal programming. So fetal growth is dependent on the placenta. So in normal placental development, trophoblast progenitor cells proliferate and differentiate into the syncytial trophoblast and cytotrophoblast that together provide an interface responsible for nutrient and gas exchange, but also hormone production and immune proliferation. And then we have the extravillous trophoblast that anchors the placenta, invades the endometrium, so we have the remodeling of spiral arteries, and adapts the uterine blood vessels to ensure optimal access to maternal resources. And when you get suboptimal placental development or function, sometimes known as placental insufficiency, it can lead to adverse complications, namely fetal growth restriction and the disorders that are associated with FGR, such as preeclampsia and maternal obesity. So, sorry. Again, a quick recap on FGR. I'm not sure how familiar the audience is, but fetal growth restriction is a condition where the fetus does not reach its intrinsic growth potential. So another description of this is basically an estimated fetal weight or abdominal circumference lower than the 10th percentile for that gestational age. So, and severe FGR would be lower than the 3rd percentile. And it's also associated with presence of umbilical artery Doppler abnormalities in some cases. And this affects about 5 to 10% of all pregnancies and is considered the second most common cause of perinatal mortality. Why are we particularly interested in FGR? It's associated with an increased risk of short-term complications, such as stillbirth and preterm delivery, but also long-term complications. So this comes in with the developmental programming aspect of my interest. So increased risk of cardiovascular disease, metabolic disorders, hypertension, just to name a few, in adulthood of those offspring that have been exposed to FGR at birth. So currently the management involves monitoring the mothers at risk and the fetuses and deciding when would be the best time for delivery, balancing the risks of things like intrauterine chronic hypoxia with the risks associated with preterm delivery. So there's currently actually no effective treatment or cure per se, which really emphasizes the need for research into potential therapies that target the underlying causes of FGR, namely placental dysfunction, and that can be things like placental inflammation or deficiencies in nutrient transport. So a little bit of background. I won't go into this in detail because I know that you're all probably familiar with extracellular vesicles, but we know that good fetal growth is associated with a maternal diet rich in fruit and vegetables, but the mechanistic basis of this is unknown, as macro and micronutrients of the fruit and veg, as well as socioeconomic confounders, does not explain the relationship. So could extracellular vesicles in these plants or these fruit and veg be the answer? Extracellular vesicles, as I said, probably familiar to this audience, is a blanket term that encapsulates several classes of cellular-derived vesicles with varying size, composition, and function. They are lipid-encased parcels consisting of various proteins, microRNAs, and messenger RNAs that can be internalized into recipient cells, delivering their cargo and resulting in altered gene expression and cell function. So just quickly to go through the process of extracellular vesicle isolation from watermelon, this basically follows the same typical protocol for extraction or isolation of mammalian EVs. So it's a series of ultracentrifugation steps. So what I do is I take the watermelon pulp or flesh and remove it from the rind, which is the skin of the watermelon, pulse that in a blender, and then sieve the resulting juice through a coarse sieve to result in a clear red watermelon juice, which you can see here. And then this is then run through a series of ultracentrifugation steps, first at 1000 G for 10 minutes, and this should result in a large dark red pellet of whole cells and small tissue fragments and a clear orange supernatant. The supernatant is then spun at 10,000 G for 10 minutes twice, which should remove any remaining large fragments of tissue, apoptotic bodies, or whole organelles, and such as plastids. The supernatant is then filtered through a 0.22 micrometer syringe filter and spun at 100,000 G for 90 minutes to isolate the extracellular vesicles of the required size. And then that's what's resulted in the pellet, which is resuspended in sterile PBS and then either used for experiments or stored at -80 degrees. So obviously before giving these EVs to mice, we actually need to ensure that we actually have isolated EVs of the correct type and size. So there's a few things that we did to characterize them before, which was measure the size using the nanosite. So we're looking for exosomes or extracellular vesicles of about 100 nanometers in diameter, in the range of 50 to 150 nanometers. We also did some transmission electron microscopy analysis to look at the morphology, and we're looking for a typical spherical or cup shape. Previous work by a PhD student in our group actually did some in vitro work with watermelon EVs in placental cells. So I wanted to repeat that before going into the in vivo model, so we can label the RNA in the EVs or the lipid. In this case, we labeled the RNA, and look at uptake into vivo placental cells. So this is just an example of some of the outputs from the nanosite. So we have 3 different preps here. We have, as a positive control, I isolated EVs using the same protocol from Ishikawa cells. So these are mammalian EVs. And then there's 2 preps from 2 different watermelons. And you can see that there is a single major peak, which shows that this is a monodispersed sample, which is what we want to see. And we can see both the mean size of the extracellular vesicle particles in the mixtures, but also the concentration, which is really important for determining dosage. So just a note that each prep uses about half a watermelon's worth of juice. And obviously, we're going to do multiple preps to get enough for dosing the animals. So as you can imagine, there'll be differences between different watermelons, but also between different preps. And you can see this when you resuspend the pellet, you can see the difference in color, which suggests a different concentration. So what we do is we actually pooled all the preps together into one master prep, which was then used for the treatment of the mice. That way we avoid any differences between individual preps. This is just an example of the images from the transmission electron microscope. So the blue lines indicate the EVs, which have this kind of cup-shaped morphology. There are some that have suggested that this morphology is an artifact as a result from desiccation of the exosomes during processing, but regardless, it's still a way of us characterizing that there's EVs in the sample. So then just going back to the work that was done by Kate Timms, a previous PhD student, so labeling the watermelon EVs that we've isolated with cytoRNA select, which should selectively bind to the RNA. We also had a dye control, which was just the sterile PBS with the cytoselect DNA dyes, which obviously will not bind to anything. And then we incubated vivo cells for 4 hours with either the PBS control or the labeled watermelon EVs. And as you can see, in the vivo cells only, or with those that have been incubated with the dye control, only the nuclei were stained with DAPI, but then with the vivo cells that had the watermelon EVs added to them, you can see that the vivo cells have taken up the watermelon EVs and internalized them, and you can see the green fluorescence with that. Just continuing with the work that was previously done in vitro. So Kate Timms was the first to show that EVs can be isolated from fresh watermelon juice, and that the EVs were internalized into an intestinal Keiko2 cell line that was differentiated into small intestine-like epithelial cells, and that this occurred in a dose and time-dependent manner. And so here we have a schematic of the Keiko2 cell monolayer on a transwell insert, and we have an apical chamber with media in it and a basolateral chamber, and basically the watermelon EVs labeled so that you could detect them were added into the apical chamber and then were taken up by these Keiko2 cells. And any alteration in what they secreted, for example, could then be captured in this basolateral chamber through the transwell. So effectively, this media in this chamber, we've called it conditioned basal media, is effectively the secretome of the KCO2 cells that have been exposed to the watermelon EVs. And what she then did was take this conditioned basal media and add it to human placental trophoblasts, so vivos and primary cells, so primary human cells. And what she found was that exposure to this conditioned basal media increased expression of syncytialization genes and increased cell-to-cell fusion in these cells. And she also did the same with first trimester placental explants, exposing them to this conditioned basal media, and showed that there was decreased apoptosis and increased extravillous trophoblast migration. So following on from the in vitro experiments, my project aimed to administer watermelon EVs to normal pregnant mice and look at effects on pregnancy outcomes such as litter size, placenta weight, and fetal size, fetal weight, and investigate potential effects on placental function. We've also started working on a further aim of trying to elucidate the potential mechanism of action, or indeed mechanisms of action, because there's probably likely more than one, and the potential role of gut-placental communication. So this brings me on to the methodology for the animal work. So wild-type C57 mice were time-mated with visualization of the vaginal plug designated as gestational day 0.5. Maternal body weight was measured throughout pregnancy, as was food and fluid intake. And then at gestational day 7.5 up until gestational day 13.5, the mice were given a daily dose by oral gavage of either sterile PBS as a control, which is what we resuspended the EVs in to begin with, and then or a low dose or high dose of watermelon EVs to get a final concentration in the circulation of either 5 times 10 to the 9 particles per mil for the low dose, or 5 times 10 to the 10 particles per mil for the high dose. And this is based on the concentrations that were used in the in vitro cell work. And this time frame was chosen because we wanted to capture whilst the placenta was still forming in the mouse. So the placenta is not established until around gestational day 10.5. And we also wanted to focus on those processes of interest that we saw were impacted in vitro. So syncytialization and migration, and they occur around gestational day 8.5 or 9 up to gestational day 11. Pregnancy was then allowed to continue undisturbed with continuation of measuring food and fluid intake, and then the mice were harvested at gestational day 17.5. So post-harvest, pregnancy outcomes such as litter size, number of resorptions, maternal organ weights, fetal and placental weights were all recorded. Fetal tail tips were collected for sex typing, and we also performed some qPCR for placental nutrient transporter expression, and then some basic placental morphology with hemotoxin and eosin staining. In terms of investigating mechanistic action, fresh fecal samples were collected just before harvest on day 17.5, and the DNA was extracted and used to sequence the microbiome. We also extracted RNA and protein from the maternal small intestines harvested at gestational day 17.5 to look at some transcriptomic and proteomic analyses as well. So just onto the first set of results, the amount of chow consumed by the experimental mice, as well as the amount of water that they drank daily, was measured throughout pregnancy and the treatment period. And as you can see from this graph, there was no significant effect of watermelon EV treatment at either dose on daily food intake or daily fluid intake. I've also collated here some of the pregnancy outcome data. So we have percentage body weight gain compared to baseline of the mums and maternal organ weight data. And again, no significant effect between, or no significant difference between the 3 treatment groups with respect to these weights. There was also no difference in other pregnancy outcome data, such as the number of resorptions per litter, the litter size, or the distribution of sexes within the litter. Fetal weight, interestingly, was also unaffected by watermelon EV treatment. Remember, this is in a normal pregnant mouse. So either dose of watermelon EV did not alter fetal weight. However, placenta weight was significantly increased in a dose-dependent manner as a result of watermelon EV treatment. So these are individual fetal and placental weights nested within the dam. So it's a nested analysis, general linear mixed model. And just for clarity, I've also plotted it as the average placenta weight per litter. So you can see that the watermelon EV groups have a higher placental weight compared to control. So with this increase in placental weight, we then went on to look at gene expression of several placental nutrient transporters. So here we have investigated the impact on glucose transport. So we have the glucose transporters, the GLUTs, GLUT1 encoded by SLC2A1, GLUT3 encoded by SLC2A3, and we also have the sodium-dependent glucose transporter sGLUT1 encoded by SLC5A1. And so analysis showed that there was no significant effect of treatment on the expression of any of these transporters. We then went on to look at amino acid transport. So in the mouse, we have 2 major amino acid transport systems, system A and system L. So here is system A, also known as the SNATs, and we have 3 different subtypes. SNAT1 encoded by SLC38A1, SNAT2 encoded by SLC38A2, and SNAT4 encoded by SLC38A4. And again, similar to the glucose transporters, there was no significant effect of treatment on the expression of these transporter subtypes as a result of watermelon EV treatment. We then went on to look at system L, which is another major amino acid transporter, also known as the LATs. So we have SLC7A5 encoding LAT1 and SLC7A8 encoding LAT2. And whilst there was no significant overall effect of treatment on the expression of either of these transporters, we did find, interestingly, that there was a significant treatment by sex interaction for SLC7A8 encoding LAT2. The p-value for LAT1 is 0.16, so not significant. But interestingly, when you plot these out separated by sex, you can see that this effect is being driven by the males. So there is a significant dose-dependent increase in expression in the males of SLC7A8. And then just for completion, I've also plotted it out SLC7A5, and I do realize that this is not significant, but there seems to potentially be a similar trend going on in the male placentas. Also looked at the taurine transporter TORT, which is encoded by SLC6A6, and again, no significant overall effect of treatment, but potentially a trend going on with the treatment by sex interaction again. And if you plot that out again, separated by sex, you can clearly see that's being driven by the males again. So to summarize this section of the results, treatment of mice with watermelon EVs by oral gavage was not associated with any adverse outcomes and caused a dose-dependent increase in placental weight. Fetal weight was not affected at this gestational day post-treatment. So It could be attributed to the gestational day of harvest. We know that in the mouse, in the last 24 hours of pregnancy, there is an exponential growth. So potentially, if we had harvested as close to term as possible, we may have seen an impact on fetal weight. Alternatively, it could be that they've reached their maximum growth potential because this is normal pregnancy, and we may have to be in a disease model of restricted growth or FGR, for example, to see any effect on fetal weight. We really don't know at this point. The expression of placental SLC7A8 and SLC6A6 was increased in a dose-dependent manner, specifically in placentas from male fetuses, which could indicate a potentially sex-dependent enhancement of placental nutrient transport function. And we hope that future work will investigate targets involving the regulation of placental nutrient transport, such as targets in the mTOR pathway. And mTOR is particularly interesting actually because previous metabolic pathway analysis on the in vitro secretome that was done by Kate, the PhD student, actually came up as mTOR, with mTOR as a potential target that could be affected by watermelon EVs. So that was some bioinformatic analysis that she did. So that would be really interesting to kind of follow that up a bit further. So now just moving on to the next aim of what mechanisms could be underlying these potentially beneficial effects to the placenta. And one of these could be the modulation of gut function and therefore gut placental communication. And a direct way that this could happen could be direct effects on gut gene and protein expression and therefore function, which could then have downstream effects on distal organs, for example, the placenta. So looking at the proteomics and transcriptomics of the small intestine, the gut in general. An indirect pathway could be through changes to the gut microbiota, which have been shown to have effects on, for example, short-chain fatty acids, which we know are important to placental function. And another possibility could also be that the immune ecosystem of the gut is being altered, and that's another mechanism that could be explored. So firstly, looking at watermelon EV effects on the gut microbiome, fecal and large intestine samples were collected from treated mice at harvest on gestational day 17.5, so these were fresh fecal samples. DNA was extracted and PCR was run against primers for bacterial 16S, and then this was then used for next generation sequences. So when we get the data back, we got it as relative abundance of bacterial populations at different taxonomic levels, and an example is shown here of the output. And we saw some significant differences at the family level in the high watermelon EV group compared to control. So we saw an increase in the relative abundance of the family Bifidobacteriaceae. I never know if I say that correctly, but we're going to go with that. So, which you can see here represented a little bit more clearly. So this enrichment of Bifidobacteriaceae in the watermelon EV treated fecal samples is really interesting because other groups have also reported Bifidobacteriaceae being among the taxa shown to be decreased in the intestines of pregnant mice fed a high-fat diet. So we see an increase here with watermelon EVs. There's also been a study by a group in Cambridge that looked at germ-free mice and found direct evidence for the ability of Bifidobacteriaceae in the maternal gut to promote placental development and function. And so they treated mice on days 10 to 14 of pregnancy with Bifidobacterium breve, which is a species of this family, and found that it beneficially influenced placental morphogenesis and nutrient transporter expression. And if you remember, we're seeing a potentially positive impact on placental nutrient transport and an enrichment of Bifidobacteriaceae as a result of watermelon EV treatment. So it kind of ties into that. These studies that I've mentioned have implicated acetate, which is a byproduct of fermentation of bifidobacteria, in gut placenta communication. And we also did some analysis using Brenda of the bacterial pathways that came up from our next generation sequencing, and they've implicated metabolism of acetate, but also other short chain fatty acids. So with some of the fecal samples that we had left over, we did some liquid chromatography, mass spectrometry analysis of the fecal samples to look at some of these short chain fatty acids, and we did find differences in the fold change, which is interesting because we know that short chain fatty acids, but also other microbiome derived signaling metabolites such as tryptophan, are known influences of placental function. So moving on to potential modulation of, or direct modulation of gut function, we collected these small intestines from the dams, and you extracted both RNA and protein for RNA-seq and for mass spec or proteomic analysis. And we found differentially expressed genes in the watermelon EV group compared to control, as well as some differences in protein expression. But we'll mainly use this data to do some pathway analysis using web gestalt. And just an example of the output. So this is for the transcriptomic analysis. We found a number of pathways come up as being predicted to be affected. And I've just circulated some of interest, which are relate to inflammatory processes or immune related pathways. So we have things like cytokine metabolic processes, type 1 interferon production, humoral immune response. And we also get the same with the proteome. So again, some pathways relating to the immune and inflammatory response here. So acute inflammatory response, for example. And this is really interesting because if you remember this slide, if I go back to this, I highlighted the immune ecosystem coming up as a potential mechanistic driver. So it really suggests that these different pathways are likely to be interlinked and not standalone. And if you remember, I mentioned bifidobacteria being decreased in pregnant mice fed a high-fat diet, but also other maternal obesity models have demonstrated intestinal inflammation. And those pregnant mice that were fed a high-fat diet also had increased markers of inflammation and ER stress in their associated placentas. So it's likely that there is a link between these different mechanisms of action. So when we saw this effect that came up on the pathway analysis from the transcriptome and proteome, it led us to look at inflammation and the immune status of the small intestine samples that had been treated with the doses of watermelon EV. So we looked at some inflammatory cell markers by immunohistochemistry. So here we have CD45, CD64, and CD3. CD45's marker of total white blood cells, CD64 of myeloid cells, macrophages, and CD3 of T cells. And what we found was that there was a significant dose-dependent decrease in the number of positive cells stained for each of these three markers, and that was across all three markers, and significant. We also decided to look at the expression by qPCR of some inflammatory cytokines, and found again a significant dose-dependent decrease in the expression of the pro-inflammatory cytokine TNF-alpha, which is here it's represented as a fold change compared to control. And this was in both the low and high dose watermelon EV. I haven't showed it here, but I also found a trend towards an increased expression of the anti-inflammatory cytokine IL-10. So to summarize the second set of results, we propose a gut placenta communication axis, whereby the mechanism of action of EVs is likely to involve a combination of factors that are likely to be interlinked, including immune pathways, the gut microbiome, and alteration of intestinal structure and function. We've shown that watermelon EVs alter the relative abundance of gut microbial populations, also alter gene and protein expression in the maternal small intestine. A future work will attempt to characterize these changes in relative abundance a little bit more, and also to characterize potential changes to intestinal cell structure, function, and barrier integrity, which is not something we've looked at yet, but we know that in maternal obesity, there is suggestion of a phenomenon known as leaky gut and changes to barrier integrity. So that could be something we could also look at. The pathway analysis from the transcriptomic and proteomic results indicated a possible role for immune inflammatory pathways. And we've also shown reduced pro-inflammatory cytokine expression and reduced immune cell populations in the small intestine. And we're currently working on the matched placentas to those small intestines to look at the same inflammatory targets in the placenta. And already we're seeing preliminary results that suggest a similar pattern of reduced TNF expression in the junctional labyrinth zone of the mouse placenta. So where do we go next from this? The aim is that future studies will investigate the impact of EV therapies in the first instance from watermelon on pregnancy outcomes in rodent models of pregnancy complications. And our choice is likely to be one to look at FGR and or placental inflammation. And one way to do that is through a high-fat diet model of obesity, because as I've mentioned previously in the slides, that is high-fat diet mouse models are associated with placental and intestinal inflammation, also have been implicated having a role of the microbiome, and produce offspring that are fetally growth restricted. So that would be potentially a good model to investigate these different mechanisms of actions. And then the hope is down the line that we may be able to determine the suitability of these watermelon EVs as a therapeutic intervention in the mouse model, but then potentially in clinical trials, giving it to pregnant women. So just thanking everyone at the Maternal and Fetal Health Research Centre at Manchester and the PI on this project, Professor Melissa Westwood, various students that have contributed, and our funding has mainly come from the Tommy's Baby charity, which is a big driver for stillbirth research at Manchester. So yeah, thank you very much, and I'd be happy to take any questions.
Thank you, Hager. And now the floor is open for the questions. So until they get ready with the questions, let me come up with the first one. So you're telling that you use the dose-dependent treatment of watermelon EVs with the mice. So how frequently did you fed the mice with watermelon EVs?
Yeah, so we gave them a daily dose for 7 days by oral gavage. So the reason why we chose 7 days is purely because of the license, and we weren't allowed to get it for longer than that. And then we chose that time window. I'm trying to go back to the slide at the beginning. Because of the reasons that I said about the placenta not being fully developed yet, and trying to capture those processes of migration and syncytialization. We did want to try and give the watermelon EVs just in the drinking water, but when we tried to work out how many EV preps I would have to do to produce enough EVs to do that, I probably would have been doing it for a year. So we thought oral gavage is probably the best way to do that. So yeah, it was for 7 days. We have been asked before, like, why didn't you give it intravenously or, as a subcutaneous injection? But because we really wanted to simulate as close to possible as ingestion, like normal ingestion, as you would eating a watermelon or drinking watermelon juice. So I think oral gavage was decided to be the best option for that.
Thank you. And we have 2 more questions. Ali Reza, you can go ahead.
Thank you very much. Sorry, and I have more than one question, if I may.
Yeah, please go ahead. We have time.
Yeah. Okay. No, it was, from several different aspects, the study that you have done is very interesting. I think probably the time that you did this study, this was probably maybe still was one of the unique studies to feed EVs to a pregnant model and somehow show that it is. I don't know if anyone has. I know with the milk they have done quite a lot, but I don't know if they have looked at the pregnant.
To my knowledge, with plant EVs in pregnancy, we are the first ones to do it, and I'm not aware of anyone else that's doing it yet.
Hence, congratulations, pioneering to look at that, and also Can I also say, these are not probably extracellular vesicles. These are vesicles. These can be all both from the extracellular and also intercellular origin. But I had this discussion with another colleague in Spain, and he said to me, I don't care where they are coming from till the time that they have function. Then whether it is intracellular or extracellular, they are vesicles, and they are doing it. But do you, then okay, the conclusion is, okay, placenta maybe is affected, but generally it looks that they have a kind of anti-inflammatory effect in then if somebody starts to give them as a kind of supplements and someone say, do this and you would have less inflammation, or if you have problem with your diet, or I don't know, generally, have you thought, or have people that when you did this work, have you come up with also that maybe, okay, we can use it as an anti, or bringing down inflammation in the body, or hoping that?
This is really interesting because the anti-inflammatory effects is very recent. So we were kind of focused mainly on microbiome and the effects on the placenta, but it was really interesting to see those anti-inflammatory effects, and they're very pronounced, and then to see them also potentially in the placenta as well. This is current work that's going on. So you're very right. It could be that, we're focusing on improvements to placental function and pregnancy outcomes, but it could have anti-inflammatory effects in the intestine or in the gut as well, which could be another avenue to explore for sure. And we've currently put a grant in to kind of focus on that a little bit more. But it could be that even if we don't see, for example, that it does rescue an FGR phenotype, you could see beneficial effects just in terms of anti-inflammation that could be added to other fields of research, not just pregnancy. So definitely, yeah, for sure.
If I can also ask one other question, did you have any other adverse effect? I mean, something that you see these animals are taking it and they are having, for example, more diarrhea, or they are more constipated, or they have any other kind of thing that, from one point, this study is showing feeding EVs to individuals as a supplement. And in this case, probably I haven't calculated, but you are feeding every day half a watermelon to a EVs of watermelon to a mice.
No, it would be definitely less than half a watermelon, because half a watermelon is producing, you're resuspending the pellet in about 250 microliters of PBS, but you're not giving that much to you're gonna dilute it down to give it to the mice, so it's less than half a watermelon for sure. But what was your question before that? Sorry?
That if you see any adverse effect, anything that you can.
This was the reason we actually did this in the normal pregnant mice first, to actually see if there was any adverse effects, and we were pleasantly surprised that there wasn't any. Like you see, it didn't affect their eating habits. It didn't affect the amount of food that they ate, or the amount of water that they drank. Their activity, like you said, their fecal, status or diarrhea, whatever, nothing was affected at all. You could not tell a difference between one that received PBS or one that received any of the doses. So that was really, that made us really happy, because it means, okay, we can continue with this and potentially go into a disease model. So yeah, so that's what we've got so far. It actually seems to, and if you remember, this is a normal pregnancy, and we're already seeing beneficial effects in normal pregnancy. So the hope is that if we do go into a pregnancy complication model, we may see those beneficial effects actually changing the phenotype potentially. One can hope. That's the idea.
And for the last one, Gulmini, sorry.
Yeah, sure. No, yes, we still have time.
This anti-inflammatory effect, was it local or was it something that you see in general blood or so? Is it something that, have you looked also in the intestine or is it systemic or is it localized?
Yeah, so we've only looked at it at this stage in the small intestines from the dams. We've not looked at it in the blood yet. We do have plasma samples collected, so we could look at it to have a better idea of like the systemic inflammatory status. But we've only looked at the intestines right now and started to look at the placentas, and the placentas seem to mirror the intestines, so they seem to have an anti-inflammatory effect in the placentas as well. But yes, ongoing work will definitely look at whether that's a localized effect or doesn't seem to be localized because, like I said, it's been in the intestines and in the placenta. But yeah, definitely to look at the blood samples is the next step.
Thank you very much. It was very interesting. And why watermelon? Why you didn't use just a melon?
I get the question all the time, and everyone's like, why did you choose watermelon? So actually, before I started on this postdoc, the idea is we don't actually think it's probably just watermelon. We probably think that some fruit and veg have a similar mechanism of action. Even if the EVs are slightly different, we suggest that the mechanism of action is probably going to be the same for a lot of fruit and veg EVs. But they started with watermelon because they initially were interested in the microRNAs within the watermelon EVs, and there was a study that found that microRNAs, when fed to, it was in males actually, they were given watermelon juice, they could see that the microRNAs were found in the circulation from these men that had drunk the juice, so that they could confirm that it was traversed the intestinal barrier and they could get these microRNAs. It's since moved on from there, and we've decided not to kind of focus on components of the EVs, but just rather their mechanism of action and them as a whole. So that's kind of where it started. We also wanted to choose a fruit that would be easy to isolate juice from, and would give you a lot of juice and a lot of EVs from each prep. So from practical reasons as well, that was why they started off with watermelon. But we do have plans to look at like other fruit and veg. For example, grapes are used a lot to isolate EVs from grape juice. So that's something maybe potentially down the line or for a student project as well.
Thank you. Very, very interesting.
Thank you.
Go ahead. Yeah, Kasun, it's your turn.
Yeah. Can you hear me?
Yes.
So have you checked about the uptake of these EVs in vivo system, where it is going, which tissues it uptakes, like does it go to the liver or the spleen, or like have you checked?
Yeah. So we did initially think that we were going to say, okay, let's harvest the maternal clearance organs and see where it ends up, and we did label them. I think because of the day of harvest, I'm not sure what the half-life of the EVs are like fluorescence, so we didn't really manage to see where they end up. But to be honest, our thinking has gone a little bit beyond whether they are, they traverse the intestinal barrier and are taken up whole into other organs. It's more, our focus has been more focused on, it's probably that the EVs are having an effect on the gut in the various ways that I've mentioned, which then has downstream effects on the placenta, rather than them actually themselves getting to the placenta or getting to other organs. That's our kind of way of thinking, and a kind of where I think some of the people in the field are also going, is rather that their effects on the gut are then having downstream effects. But yeah, you're right, that's something that we could potentially look at in the future a bit more.
And when you say having a potential effect on placenta, I'm not suggesting that it actually goes to placenta,
Okay.
but it goes to the gut. And do you think it is the nutritional effect, or like EVs are taken by the gut and there are microRNA is doing something in the gut and then it affects the placenta? Do you have a hypothesis at this moment?
Yeah,
to how your system works?
Still forming. So I tried to maybe suggest some things that are going on. So whether it's, you know, whatever it is in the EVs that's altering the gut cells function, we've seen that it is affecting the transcriptome and the proteome of the small intestinal cells. So it is having some effect. You know, it could be then this effect has an effect on the microbiota as well, which then alters what they release and their byproducts, the short chain fatty acid, which could then have downstream effects. So I think that's probably what we're thinking. It's probably these EVs, whatever's in them as well, is potentially having an effect on the gut in various different ways, whether it's altering the immune ecosystem of the gut, or it's actually affecting the gut cells themselves, the transcriptome, the proteome, and then basically whatever they secrete, and or having an effect on the microbiome. So this is kind of the three mechanisms that we're looking at. I hope that answers your question.
And finally, have you tried this with any other kind of EVs?
No, not yet.
It's just watermelon. What is your control here?
We have a student project that was working on grapefruit, so we're potentially going to see if we can isolate AVs from grapefruit as well, and see whether we can whether they have a similar profile or whether they have a similar effect as well. Yeah.
Thank you very much.
You're welcome.
Yeah, and this one last question. Yeah. So with your microbiome data, we can see that the bifidobacterium has been increased, but then the bacteroides gone down.
Yeah.
Yeah, so meaning that it has a kind of antibacterial or some kind of selective toxicity. Yeah. At least I can see it. They can see it in the bacteria. So did you check it for like relative the toxicity for the
no,
tissues or?
Really good suggestion. No, not yet. I am not a microbiologist, so this is all new to me, but I've been trying to read up on like the ratios of certain taxa families as well, and that means it's antibacterial and probiotic and so on and so forth. But yeah, definitely something to look into. So a lot of the data that we got from the sequencing of the microbiome data, we used to kind of do the pathway analysis to inform what we can do next, but definitely want to drill down a little bit more on the differences in the relative abundances and what that means and what the different families mean and so on. So definitely something to look for in the future. Yeah,
because in your data I've seen that lactobacillus has not been changed, like hardly changed their percentage, but then the bifidobacterium specifically gone high with the high watermelon EV dose.
It's really interesting because, like I mentioned, that work that was done by Cambridge Group on bifidobacterium, and that was all came out around the same time as well. And they specifically were looking at placental function, and they were in germ-free mice. So that was interesting that we got a similar enrichment as well. But yeah, definitely the other groups or the other species to look at and see what that means would be interesting to look at. Yeah, and I only showed one example, but there are many graphs to look at and loads of different families and species to look at as well. So,
Andres, please go ahead.
Yeah, how expensive or how hard is to do this? Because we have now several sources of EVs. We make also our EVs to become lyophilizers, hence it is easier to store them. But we have them from milk, we have them from plants like potato, we have them from the algae and so. But to set up these experiments, if we want to set up such experiments, how many mice you need to be able to, I mean, what was the minimum number that you were needed? And so, and also if you guys are interested to have, if we provide the EVs, is that something that if it is set up in your system, would you?
Now when you said potato EVs, I got perked up because I got very interested in that, because if you've already isolated them from them and characterized them, that would be really interesting, because we've now done a fruit and we'd really like to do a vegetable next, so it would be really good to potentially do that, and I'll definitely have that discussion with Melissa and let her know you're interested, because yeah, why not, for sure. In terms of numbers, so we still consider this pilot data because I said it was mainly, but we managed to do a bit more than we initially expected to do. So we had about 7 to 9 in each group. And we did 2 doses to begin with because we had no idea how they were going to respond and what would be the right dose. Probably going forward, we'd probably just choose one of the doses, probably the low dose, because it's both having an effect. So that's why we may have needed a bit more numbers, but I would probably try to aim for maybe 7 to 9, 7 to 10 in each group. That's what we saw effects with, significant effects with. But yeah, that's
Yeah, we are producing quite a lot of different ones, and we have the EVs. If you guys, since you are also experienced, if you are interested, we can easily and as I said, we lyophilize them. Hence, you don't need even to store them in -80. It would be coming in. Milk EV, we have it even commercialized. We somehow sell it. But the potato we can definitely we can supply. That can be a collaboration we can set up.
Yeah, definitely. Yeah, thank you.
Is there any more questions to Hager?
Yes, I can. If I have
Yes,
only a
yes,
couple of
yes,
questions.
Gaetan. Please go ahead.
Yeah, it was a really nice presentation. But my question here on this slide, you only use 2 concentration of EVs. Have you tried to, I don't know, to go to add more, like to the right or to the left? Like, why only use 2 concentrations?
It's just feasibility wise. I mean, you don't know how long it takes to make enough EV preps to give into because it's not the same as in vitro work. Obviously, like you'll need a lot more to give to the mice. So working out the concentration, the final concentration, and the circulation. So we actually used these 2 doses because we based it off the concentrations that we used in vitro, and Kate had used a higher dose, so a tenfold difference between them, and that's why we just started off with those doses to just see if we at this point we had no idea. No one had ever fed EVs to pregnant mice, so we didn't know what we were going to get, so we chose what we designated a low and a high dose. And for practicality, adding more than 3 treatment groups would have probably been difficult in the first instance. Now that we have this data, we could probably say, can we go lower and see an effect? And if we go higher, we're probably going to see an effect because we've already determined that. Can we go lower and see an effect with a lower dose? We could potentially do that, but I think we're probably happy with deciding on one of these doses and kind of looking at more aspects of the phenotypes rather than kind of looking at different doses.
Yeah, but another thing also, the way you introduce these EVs are based on oral, right? Like oral gavage. Yeah. But have you tried different way? Because if you put orally, it goes to different sites. And I don't know if your EV survives this site, different location in the system.
Yeah, so again, I always get this question about why did you do oral gavage? Why didn't you inject them, etc. And again, I'm going back to the aim down the line is to give this to pregnant women, right? And you want the best mode of delivery, which is, and it's, again, we're developing dietary therapy, so we're trying to do it in the most natural way possible, which is probably going to be ingestion. So that's why we've gone with oral gavage. And again, we wanted to look at the effects on the intestine, the gut. So we want that digestion, we want that to go down to the gut. If you do it injections subcutaneously or intravenously, you're not going to get that. So that was our reasoning for using oral gavage rather than another mode of delivery. I know that there have been people who have used injections and other modes of delivery, and that may be able to show you sort of more localized effects or direct effects, but this is why we use this in this instance. That makes sense. Yeah.
Fine. Yeah. Okay. Thank you.
Injection would be completely another can of worms. That they would think about the septic situation if these EVs are themselves produce quite a lot of problems with combat. But can I ask, these 5, for example, the high dose 5 to the 10 power of 10, is this exactly in the water that they are drinking? You have that concentration?
So we work out basically weigh the mouse, work out the blood volume for that mouse, and then basically like what we give them by oral gavage is actually a higher dose, so that by the time it gets to the mouse, that final concentration is about that. 5 times 10 to the 10 particles per ml.
Per ml of the drinking water or no?
So it's off what they're actually given by Olga Vaj, so in the actual syringe.
In the actual, and how many milliliter, I mean, per day they are?
It's like less than 500 microliters, less than,
That they, okay.
So yeah, if there's no more questions, Dr. Hager, thank you so much for your insightful presentation, and we're looking forward to make some collaborations in future. And thank you very much, and all the best for your future work.
Okay,
thank you, everyone.
I appreciate it. Okay.
Thank you. All the best. Thank you.
Bye.
Thank you.